Hi, welcome to the 6.4 control of blood water potential video.
Specification.
It's not very long, but you can pause it and read it if you want to. Okay, so up here we have a diagram of the kidney. Now, the reason that this is in there is because I just want you to be able to see the fact that this here is what we call a nephron. So these small structures are placed here within the kidney. And obviously you've got loads of the nephrons all the way around the kidney. You've also got blood coming into the kidney via the renal artery, red here, that's bringing blood in that has waste, and then you've got blood leaving through the renal vein. This here is your nephron. So you can see here it's turned on its side. The parts that you need to know here, you've got the afferent arteriole that is, in fact, let me just pop that there. That afferent arteriole is bringing blood into the glomerulus. So the arteriole is just a branch of the renal artery. This is a little kind of bundle of capillaries. So blood comes in the afferent arteriole and it leaves, and this is called the efferent arteriole. It doesn't have it labelled there, but it's the efferent arteriole. So if you remember afferent, A for afferent and A for arriving, and efferent is E for efferent and E for exiting. So this is the blood arriving into the kidney, into the nephron, and then leaving. Then you've got this area of the nephron here. That's the Bowman's capsule, and that surrounds and kind of holds the glomerulus. And then the nephron continues. So the nephron's like a big long tube. This part of the nephron here is called the proximal convoluted tubule, or the PCT. So that's your PCT there. This bit here is the loop of Henle, all the way up here. And then you've got the distal convoluted tubule here and the collecting duct. And then once, what happens is there are molecules and water that are squeezed from the blood into here, into the Bowman's capsule, and that filtrate moves all the way through the nephron until it gets to the end of the collecting duct where it is urine. And that urine, if you can see on this diagram, collects here in the renal pelvis and then moves via the ureter to the bladder. And also later on we'll talk about ADH. And ADH is the hormone that controls the concentration of the urine. So sometimes sometimes the urine is dilute, sometimes it's very concentrated. So ultrafiltration occurs in the glomerulus and the Bowman's capsule. So we said that the blood enters through the afferent arteriole and leaves through the efferent arteriole and flows through this kind of mesh of capillaries. The blood within these capillaries here is under really high pressure, so it means that the components of the blood can be squeezed out, and when they're squeezed out, they're squeezed into the Bowman's capsule. So there's really high pressure within these capillaries, and it squeezes the molecules into the fluid in the Bowman's capsule, which we refer to as the filtrate. We've done that one. The molecules have to pass through. So if you look down here, this is the capillary within the glomerulus. The molecules have to pass through the capillary wall. They have to pass through a basement membrane of the glomerulus. And they also have to pass through the glomerular membrane. These here are special cells called podocytes. You don't need to know that for A level, but that's what they are. And you've got the capillary fenestrations here. It just means kind of gaps between the cells within the capillaries so that things can squeeze out. The podocytes help to kind of filter the molecules that are moving into the filtrate that's here. So you've got capillary wall, basement membrane, glomerular membrane, and the filtrate. Now, things that pass from the blood into the filtrate are water, ions, glucose, and urea. And plasma proteins cannot pass from the blood into the filtrate because they're too large. They can't get through these fenestrations within the capillaries, so the plasma proteins don't move into the filtrate. So that's the process of ultrafiltration occurring here in the Bowman's capsule. We've then got selective reabsorption, which starts within the proximal convoluted tubule, and the clue is in the name. So it's reabsorption of some of these molecules that made it into the filtrate. So some of them are going to be reabsorbed. So you can see that the proximal convoluted tubule is surrounded by a network of capillaries. So what's going to happen is some of the molecules are going to move from the PCT back into the capillaries. And the walls of the proximal convoluted tubule have microvilli in order to increase the surface area, because obviously if you increase the surface area, more reabsorption can occur. So all glucose that has moved into the filtrate is going to be reabsorbed in a healthy person. Obviously, if they have diabetes, some glucose may make it through into the urine, but in a healthy person, all glucose should be reabsorbed. Some water is going to be reabsorbed and some ions will be reabsorbed as well. And the water is reabsorbed obviously by osmosis, because water only ever moves via osmosis. And the water potential is higher in the filtrate than in the blood, which allows the osmosis to occur from the inside of the PCT into the capillaries. And obviously, whenever water moves by osmosis, ions follow, so that ions will follow the water back into the capillaries as well. Okay, the loop of Henle. So the loop of Henle is this bit here. And there's an extra diagram here for you to look at. It's quite handy because it's got some numerical values which might help you. So you've got the descending limb of the loop of Henle, the bit that goes down, and the ascending limb, which is the bit that goes up. Now in the descending limb, water is reabsorbed from the filtrate into the medulla. So the medulla is just the area of the kidney that the loop of Henle is found in. So the water is reabsorbed into the medulla. It will eventually then move into capillaries, but just in this little area, it moves into the medulla. So you can see that you've got water moving out. Now these numbers refer to the solute concentration within the filtrate. So obviously it's getting more and more concentrated as you move down because the water is moving out. And this is going to be your most concentrated area of filtrate within the whole of the nephron. In the ascending limb, where we're moving up, you can see that you've got chloride ions and sodium ions moving out. They move out here by passive transport because there's a higher concentration. However, when we move up to here, there's a lower concentration, so they have to be actively transported out into the medulla. The ascending limb of the loop of Henle is not permeable to water, so no water moves out by osmosis. That only happens here. So that's the loop of Henle. So you've got water absorption, descending limb, sodium chloride absorption, and the ascending limb. And then in the distal convoluted tubule, more reabsorption occurs, and this reabsorption is controlled by hormones. And then just some added information, the fact the kidneys need and the nephrons regulate the water potential of the blood. And so when there's too much water in the blood, more dilute urine is produced. And when there's not enough water in the blood, concentrated urine is produced. We'll look at that in more this slide here. So, this is really useful actually. There's lots of exam questions that are like 5 and 6 markers for this topic. So describing ultrafiltration for 5 marks, or describing selective reabsorption for 5 or 6 marks, or describing the control of the how ADH affects the collecting duct could also get you quite a lot of marks. So osmoreceptors in the hypothalamus in the brain regulate water potential of the blood. So the blood is always moving past these osmoreceptors, and what happens is if the water potential of the blood is low, water will move out of the osmoreceptor cells in the hypothalamus, and this will then trigger the posterior pituitary in the brain to release ADH into the blood, and ADH is a hormone. The ADH moves to the kidneys, and its job is to make the collecting duct walls more permeable to water. Now, this means that obviously when we release the ADH is increasing. So therefore, there's a lower blood water potential. As we said, it causes the collecting duct walls to become more permeable to water. So therefore, more water is reabsorbed from the urine, and the urine becomes more concentrated. If we decrease the ADH, it means that there must be a higher blood water potential. So no ADH needs to be released. The collecting ducts therefore become less permeable to water, less water is reabsorbed from the urine, and it becomes more dilute or will be far lighter in colour.